let me first start by saying thank you so much to Christine for inviting me to give this talk. It's a pleasure to be here with you guys today. This is a treat because I've never been to Whitney before, and I've seen a lot of it on videos. I was just telling Christine I saw a video when Dr. Associate Provost Glover came down here, and I really enjoyed that video. So I get to be here now and see more of Whitney, and I'm really happy to be here with you guys as well, to share a little bit about my work. But before I get into my work, I will first and foremost tell you a little bit more about me. You might hear a slight accent, and that's because I am from the Caribbean. I was born in Guyana. Anybody know where that is? No? Yeah? You've been to Guyana before? No? Oh, gotta go. It's actually probably the latest oil-producing nation in the world. So, you know, if I ever quit this, you guys know where I might be. But I actually grew up in Jamaica. I'm pretty sure a lot of you have heard of Jamaica. Anybody ever been to Jamaica? Vacation, I assume? Yeah, so I grew up where you guys vacationed. But then I came here for college. I went to Florida A&M University, Rattlers. Anybody heard of Rattlers? It's one of the HBCUs in Florida. Then did a master's in biomedical engineering at UNC North Carolina State Biomedical Engineering Department in Raleigh and Chapel Hill. And then came back to Florida to do my PhD in biomedical engineering here at the University of Florida. My project, which I'll tell you guys a little bit more about it in my talk. My project got really interesting, and so I decided to stick around and continue doing a postdoc at the University of Florida. And then that, after a year, turned into actually a company, right? So my PI, myself, and a couple other immunologists started the company, which was called OneVax. And so that made me stick around till about 2015 in Gainesville, after which I decided I kind of wanted to do my own thing, pursue my own ideas. And so I decided the best place for that would be academia. And so I started my own lab at University of California, Davis in 2015. And I was there up until 2022 when, you know, the lure of Florida, the lure of University of Florida, the lure of family and friends sort of brought me back to Gainesville. And I've been here since. I'm now an associate professor in biomedical engineering, and I just assumed the role of graduate coordinator, which I'm starting to regret. But yeah, let's get into it. So before I actually start to talk about what the projects that I'll share with you today, I wanted to pose a question to you guys. And the question is, what, in your opinion, is the ultimate nanotechnology? Anyone? And honestly, I don't know the answer to that question either, right? But just to get you guys thinking about nanotechnology, right? What? Pardon? Inject a cure. Okay. Anything else? Any other ideas come to mind when you think of nanotechnology? Anything come to mind when you think of just nano? Yeah, small things, right? What are some of the small things that we find in our bodies? Cells? The cells are still kind of big, right? What else? What else? Germs, yeah. What are some of the germs that we find in our body? Somebody said viruses, right? What else? All kinds, viruses, yeah, all kinds. What else are in those all kinds? Somebody said bacteria? Yeah. And anything else? Fungi, yeah. So we're going to talk about all those things today because my lab kind of works at the interface of all of these things, right? So in my opinion, right, yeah, pathogens, particularly viruses, some people may differ, right, in their perspective, are currently the ultimate technology, right? And I say that because, well, we spent the last 5 years being essentially traumatized by a virus, right? Yeah, yes? You can't hear me? Oh, okay. I'll try to project as best as I could. Oh yeah, sure. That's a good idea. Better? Okay. Yeah, so what I said is that, maybe a little bit too loud. In my opinion, right, the ultimate nanotechnology, at least at the moment, are actually viruses, right? Because we spent the last 4 or 5 years being essentially shut down by a virus, right? The SARS-CoV-2 virus, okay? And believe it or not, these viruses, these bacteria, these fungi, right? They are the organisms that really inspire what I do, right? And what people of my ilk, which we call immunoengineers, right? So it's sort of a new field of engineering, things you don't really consider engineering. But what we call immune engineering or immunoengineers, these little bugs, these little germs, little viruses, fungi, bacteria, they inspire exactly what we do. And what we do is that we try to essentially mimic these really small organisms, pathogens, germs, right? To develop technologies, develop nanotechnologies that can imitate them, interact with your immune system, right? But then drive changes in your immune system that are beneficial to you, to your health, okay? And so that's essentially the goal of what people like myself do and what I'll share with you, some of what I do in this talk, is that we essentially use synthetic things, right? Things that we can make, materials we can make, biomaterials is what we call them, right? To generate nanotechnologies, microtechnologies that look like viruses, look like bacteria, look like fungi, right? So that we can then put that back into your body, have them interact with your immune system, right? And reprogram your immune system, right? To then execute some sort of health programming, okay? So things that are beneficial for you, unlike the virus, okay? And there's probably no better example of this. I'm showing this slide, which I actually take from one of my classes, right? But in some of the things we mimic, right? We mimic the size, right? We mimic the shape of bugs, right? We mimic some of the molecules they present to your immune system, okay? Just so we can generate, number one, interactions with your immune cells, number two, changes in your immune cells, right, that are beneficial to your health. Okay, so that's the main thing I want you to think about as we go through some of these talks, okay? And some of the things that we particularly target, right, we target what are called APCs, antigen-presenting cells. And you guys in the last 5 years would have heard a lot about these cells, right? So these are things like dendritic cells. Anybody ever heard of dendritic cells? Right, so during the height of the SARS-CoV virus, there was a lot made about dendritic cells, because those are the cells that actually are important for generating responses to things like vaccines, okay? So we try to target those cells by mimicking shape, mimicking size, mimicking certain molecules that viruses or fungi might present. We try to get things to your lymph nodes, okay, which is sort of the command center for your immune system, okay? We try to design things that can penetrate mucosal barriers, right? So, for instance, things you can ingest and just be taken across your digestive system into your internal systems. We try to design things that penetrate tissue barriers, right? Things that target specific immune cells, and then things that target specific organelles, right? So your cell is an entity of itself, but your cell has subdivisions, smaller parts, smaller compartments. And so we try to design things that can actually get into those compartments, because they're actually important for driving what the cell ultimately does, okay? And ultimately what your physiology does as well, okay? And we do this, again, by designing particles, right, that are particular size, particular geometry, particular hydrophobicity, right? Does it interact with water or not? We put different molecules in these particles as well to drive responses in the cells to these particles. And then we also functionalize the surface of these cells to drive that interaction with immune cells. And, you know, whenever people ask me, well, what do you do? Well, the best example I could really give you for what we do is we do something similar to what the COVID vaccine does, okay? Which is simply package molecules, biological molecules that can code cells, code immune cells, right, to benefit your health into materials, okay? So SARS, we do, you know, you guys would have heard a lot about lipid nanoparticles, I'm pretty sure, for the last few years. We do some work with lipid nanoparticles, but we also do some work with biomaterials, with degradable biomaterials, right, which we'll get into in a second. Now, we do a lot of work with immune cells and immune system. That's our focus, right? But we're not trying to counteract infectious diseases in my lab. What we focus on are autoimmune diseases, okay? And then we've also moved into neurodegenerative diseases as well, okay? So I'm just giving you guys a preface to sort of educate you on what I'll bring up in this talk. All right, so my lab at the University of Florida is called the Immunomodulatory Biomaterials Lab. It's a mouthful. But as the name suggests, right, we try to modulate your immune system for the benefit of health. We try to modulate your immune system. We try to develop new systems, particulate systems, right, which we call immunotherapeutics, right, that can modulate autoinflammatory diseases, right? And in doing so, there are a couple different wings to the lab. The first wing is focused on particulate immunotherapy for autoimmunity and allergy. And I will share one story from that wing of the lab in a bit. The second wing is focused on something called vomocytosis, exploiting vomocytosis, deciphering vomocytosis. Most of you are probably like, what is that? You had your question, you had your hand up? So, have you ever heard of phagocytosis? Yeah? Yeah, cells eat, and cells eat particulate materials, they eat viruses, they eat bacteria, they eat fungi, right? And that's called phagocytosis. Okay, that's a pretty popular behavior that's probably been studied for 70, 80 years, okay? There are some cells, there's some special fungal cells and bacterial cells that when they're eaten by what we call innate immune cells, macrophages, dendritic cells, monocytes, right? They're actually able to go into the vacuole, what we call the phagosome of these phagocytic cells, okay? And they're able to survive in there for an extended period. They're actually able to multiply within these macrophages or dendritic cells. And then they do something really weird, right? Actually, they do something really weird before that, but we'll get to that. Yeah, they're actually able to get those macrophages to vomit them back out. But the crazy thing is the macrophage survives. Yeah, and those fungal cells survive, right? But yeah, that behavior of them vomiting, right, or ejecting those cells back out is called vomocytosis. And we're going to talk about that in a few as well. I'm going to bring you one story from my lab on vomocytosis. We also do a lot of basic science trying to look at cancer metabolites and their impact on immune cells. And that's because we reverse these cancer metabolites to generate new materials, new particles that can treat autoinflammatory diseases as well. Okay? All right. Everybody still with me? Yeah, okay, great. So the first story I'll bring you is from the particulate immunotherapy and autoimmunity, particulate immunotherapy for autoimmunity and allergy wing of the lab. And that story is focused on rheumatoid arthritis, right? So rheumatoid arthritis, I'm pretty sure you've heard of it before. That is what we consider an autoimmune disease. It's a really debilitating autoimmune disease, right? Where essentially your own immune cells, right, your own immune system sort of attacks your joints and degrades your joints, okay? It's a chronic, very debilitating disease, right? And if left unchecked, it can cause serious problems, okay? So it will first and foremost degrade your joints, right? So you see people with rheumatoid arthritis, right? Their hands are very swollen, their joints are very crooked, and they're in a lot of pain, okay? They're in a lot of pain. And if that pain is not addressed, right, eventually you'll be out of work, okay? So this has serious socioeconomic consequences if not addressed. And if even left further unchecked, it results in systemic inflammation, okay? So we're talking beyond rheumatoid arthritis, osteoporosis, right? Systemic kidney disease, right? So if you really don't treat it, it really envelopes into something really bad and eventually death, okay? So in my lab, we're interested in finding ways to treat this disease. Although currently there are relatively effective treatments out there, right? So currently, if you were to be diagnosed with rheumatoid arthritis, right, you'll probably go to a clinician and they'll be like, hey, you got rheumatoid arthritis. Don't worry, we can keep this in check. We'll put you in some methotrexate and a glucocorticoid, right? Maybe prednisone, okay? And they'll give you a combination of those drugs typically. And in about 40% of people, that works, okay? But in the other 60, it doesn't work, right? And in 40% of the 40 that works, right, a year later, it stops working, okay? So we have a huge patient base where this is ineffective. And so you go back to the doctor and be like, doc, this is not working, okay? Give me something better, right? And they'll say, okay, we'll put you on what we call a BMARD, right, a biologic. Okay, and so these are things like rituximab, infliximab, humara, right? These are what we call antibodies that actually target different immune mediators. Okay, so tumor necrosis factor, that's an important molecule for generating immune responses, inflammatory immune responses against things like COVID. Okay, so you get on these BMARDs, right? Things like rituximab, and you guys would have seen commercials for things like rituximab, right? Humara as well, right? These commercials are on the TV every day. You see people running through fields, being all happy and merry. And then in the middle of the commercial, it kind of goes silent, right? And there's somebody just whispering a bunch of stuff, right? You ever wondered what they're whispering? Pardon? Disclaimers. What do the disclaimers say? Pardon? Yeah, exactly, right? And with respect to these drugs, right, methotrexate, all of these drugs, which we either call DMARDs or BMARDs, right, they suppress your immune system, right? Once you suppress your immune system, it means you're not susceptible to infections, okay? In this day and age, do you want to be susceptible to an infection? COVID flying around, RSV flying around, flu getting worse, right? But not only that, your immune system is critical for monitoring and eliminating cancers. Okay? So what happens with these patients that get on these immunosuppressive drugs is that they have high susceptibility for infections, right? Upper respiratory tract infections, right? Number two, they have a high susceptibility for malignancy. Okay? So we're like digging a bigger hole to fill another hole. Okay? So they're effective drugs, right? But there's still serious inefficiencies with these types of drugs. And so we are trying to address those inefficiencies by coming up with methods that specifically target the disease, okay? Just rheumatoid arthritis, okay? And not suppress your entire immune system, right? Or wipe out parts of your immune system, okay? The thing with these BMARDs, right? So humara is an anti-TNF that'll wipe out your tumor necrosis factors, which are important for generating immune responses, right? Rituximab is anti-CD19. CD19 is on all of your B cells. B cells are pretty important for generating antibody responses, okay? So if you wipe out those B cells, it's a problem. It's a serious problem, okay? And so what we hope to do instead is target the cell that initiates this disease, initiates that inflammatory cascade within your joints, okay? Initiates the degradation of your joints. And that's the dendritic cell, the same cell that's pretty critical for vaccination, right? It turns out it's the same cell that's pretty critical for maintaining homeostasis, immune homeostasis in your body, maintaining and ensuring that your immune system doesn't attack your own proteins, doesn't attack your own cells, okay? And what happens from an immune perspective in rheumatoid arthritis is that dendritic cell, right, it initiates the attack. It'll present antigens, present those proteins to T cells, activate those T cells, right, which will then start this inflammatory cascade, which will just wipe out the cartilage in your joints and then wipe out the bones in your joints as well, okay? And that's what leads to the tremendous pain and suffering that these patients experience. Our hope, though, is to reverse the phenotype of these dendritic cells, right, that are presenting antigens or proteins that are relevant to the joints, only the joints, okay? Because we don't want to present proteins that are relevant to your muscles or your gut, right? Because we don't necessarily want to suppress immune responses to those proteins. We want to suppress immune responses to just things in the joint where we're having those inflammatory or autoinflammatory reactions, okay? Does everybody follow me there? It's a little bit of immunology, so I just want to make sure everybody's still with me. Okay. All right, and so we published on this in 2019 in ACS Applied Biomaterials. And exactly what we did is that we developed this microparticle-based system. Okay, these are microparticles about, well, I can switch to the next slide. There are about 4 different microparticles, right? First and foremost, these microparticles are made of a material called PLGA, right? It's polylactic glycolic acid, okay? Lactic acid and glycolic acid, that's it. It's just a polymer of those 2 molecules. But it's a beautiful polymer. It's an FDA-approved polymer, and it's made up of these 2 molecules that already exist in your body, right? So it's biodegradable, and it's safe. In fact, has anybody ever had sutures? Yeah, that's one of the materials that they use for sutures, okay? So it's a really, really versatile material. Can be used for sutures, can be used for tissue engineering, can also be used for drug delivery, okay? And so what we did here is that we made these particles made from this PLGA, polylactic glycolic acid, right? Lactic acid. Everybody's heard of lactic acid, right? Stuff that builds up in your muscles when you run too much or something like that. Exercise too hard. Loaded this polymer, made this polymer into different particles, right? 2 particles that were what we call pretty large and non-phagocytosable, so they couldn't be eaten by your macrophages, your dendritic cells, your innate immune cells, right? And 1 micron particles, right, that could be eaten by your macrophages, your innate immune cells, okay? And in those smaller microparticles, the 1 micron particles that could be eaten, we put vitamin D3, which it turns out is a pretty, really strong suppressive drug, immune suppressive drug, and collagen 2. So collagen 2 there is what we call the antigen, or the protein. It's the major protein within your cartilage in your joints, okay? And so the idea, before I get to the idea, the other 2 particles, the large particles, okay, they encapsulated TGF-beta1, which is an anti-inflammatory cytokine, okay, and GM-CSF, which is something called a chemoattractant. So it can actually draw dendritic cells, draw macrophages to wherever we inject them, okay? And the idea here was that we could take this microparticle system, right, just like if you get a scratch or something like that, inject them onto your skin, okay? The GM-CSF, because it's a chemoattractant for these immune cells, will draw the immune cells into that injection site, okay? Once those immune cells are into that injection site, right, they would start to eat the smaller microparticles, right? They would also be exposed to TGF-beta1 because, again, the material is biodegradable. Once we inject it, it's biodegrading, it's releasing its contents, okay? So it's releasing that TGF-beta. Those cells come in, they experience that TGF-beta1, they start to become anti-inflammatory. They also eat that small microparticle containing vitamin D3, right, which makes it even more anti-inflammatory. But they also pick up that small microparticle containing the antigen. And that's the important bit, okay? Because now they have the antigen that's specific for rheumatoid arthritis, the protein that's specific for rheumatoid arthritis. They can process that, they can present it, right? But instead of presenting it in an inflammatory mode to your T cells and your B cells, they're gonna say, hey, T cells, B cells, this is actually good protein, okay? And in that way, it'll stop your T cells and B cells from attacking the protein in your cartilage, okay? And once it stops that attack, it means we stop that inflammatory cycle of degradation in your joints, okay? And that's how we reverse this disease using this system, okay? At least that's how we hypothesized. But our hypothesis here turned out to be true, right? I'm gonna show you a little bit of preliminary data. These are what the particles look like by scanning electron microscopy. This is just a little bit evidence that we were able to load the particles, load the materials, load the drugs into these particles. And use this pointer. And also look at, the release kinetics of those drugs from the particles. And then we actually put these particles into mice, right? There are a couple established mouse models for rheumatoid arthritis. This is called the CIA-FIA mouse model that we used here. And what happens is that we actually induce these mice to become arthritic, to become rheumatic arthritic. And once they become arthritic, their paws get really swollen, they get really red. And we can assign a score to how red and how swollen those paws are, okay? And so we allow those mice to become arthritic to a score of about 6, right? And then we start to inject our treatment, which we call RegVac, right? So it's called RegVac because it's a regulatory vaccine. Instead of your conventional vaccines, we're flipping the switch here, right? Trying to generate anti-inflammatory responses. And so it's called RegVac, regulatory vaccine. But once we start to inject our regulatory vaccine, right, what we see is that the scores, this is the blue line, they go from 6 back down to 0, okay? And so that's from being what we call moderately arthritic, right, in real human patients, back down to non-arthritic at all, okay? Really strong evidence that we could reverse the disease. We did a whole lot of other stuff, which I'm not gonna talk about due to the interest of time. We do histology to confirm that. But we also look at the immune system and see if we're modulating the immune system using our microparticle system. And what we see is that when we inject our RegVac into these mice, we're able to generate what we call regulatory T cells, okay? So these are T cells that typically put a halt to inflammatory immune reactions, okay? Put a halt to inflammation. We're also able to generate what we call PD4 T cells, which are another subset of T cells that are able to sort of halt or abrogate inflammation. All right, so really quickly, these are what the mice look like essentially before and after injection with our microparticle system. So you can see initially their paws are really swollen. You can see they're very gentle under. In this paper, we did a lot of work. We actually looked at things like their mechanical gait, so looking at their stride length, their speed of locomotion, their paw force, a number of different mechanical gait analysis to confirm that, yeah, we're actually modulating the disease and improving these mice, bringing them back to being in a healthy condition. All right, so where do we go from here? Well, we've actually applied the system to a number of other autoimmune diseases, okay, at least in mice. We have applied it to early or preclinical diabetes model. We've applied it to a late diabetes model and actually shown some reversal of diabetes or type 1 diabetes in mice. We've applied it to multiple sclerosis, or a model of multiple sclerosis, advanced multiple sclerosis. And we've also applied it to rheumatoid arthritis, okay? That's all to say that in a number of different indications, this system has worked, okay? And so because of that, right, that really motivated us to commercialize this technology, okay? And so in 2013, fresh out of my PhD, I started a company with my previous mentor, Ben Kozlowski, and a couple other immunologists, Mark Atkinson and Todd Brusco at the University of Florida. That company was called OneVax, and the effort there was to sort of get this technology through the FDA and hopefully into patients. We actually did pretty well initially. We had a number of what we call phase 1 SBIR grants, which helped us to keep the doors of this company open and keep the research going and keep figuring out or keep pushing the technology towards the FDA. And then we actually also got, in 2019, we then converted to, 2020, we then converted to an S Corp. And so the company was called Inspire Therapeutics. And then we were also lucky enough, or fortunate enough to get a couple phase 2 grants to keep moving this technology forward as well, for about $2 million, to the tune of $2 million. That being said, unfortunately, we've had to shutter our doors. We had to shutter our doors last year, January, because we ran out of funding. So if you guys are interested in, or know anyone interested in funding this technology, passionate about rheumatoid arthritis or other autoimmune conditions, we have a great technology that, at least I believe in, and my co-collaborators and co-conspirators in this technology believe in. And honestly, we continue to innovate on, right? This is 2019 work that I've been showing you. We have improved the system since 2019. I just can't show it to you as yet. And so, yeah, if you're interested in knowing more about this technology, this company, feel free to contact me, or even we can chat after this talk. Should I hold the questions for this, or just, okay. All right, so I'll move on to one of the other projects in my lab. And that is looking at vomocytosis, right? And one of the other stories emanating from my lab, and that's focused on vomocytosis. Has anyone ever seen this show or heard of this video game? Yeah, you've seen The Last of Us? Anybody else? I saw a hand go up there. Yeah, you saw the entire show? Yeah, enjoyed it? Did you see this scene in the show? No. So this was actually The Last of Us is a show that came on HBO in 2022, in essentially the height of the pandemic. And I don't want to be a spoiler, but it's a show on zombies, right? It's a really good show. I think it won a bunch of Emmys last year. But this is the first scene from that show. And in that scene, there is a virologist, a mycologist, and a host. They're sitting having a conversation about, what is the next existential threat to the human race, okay? And the virologist goes off on a spiel. He's like, it's going to be viruses. Viruses. Ice caps are melting. Viruses are going to be released from those ice caps, and they're going to wipe us out. The mycologist says that's BS. That's malarkey, okay? And he's like, it's going to be fungi, right? If you think about it, right, where do all sort of neurotic drugs come from? Neuropsychotic drugs come from? Fungi, right? And then he gives the case of the zombie ant fungus. Anybody ever heard of zombie ant fungus? Yes? No? I see a few heads shaking. So the zombie ant fungus is a really interesting fungi, because it's a fungi that it can actually infect ants. And this exists in real life, by the way. It can infect ants through its spiracles, and then actually get to the brain of the ants and control the ant, direct its actions until it ends up consuming it, okay, or it ends up essentially consuming it because it multiplies in it, okay? And so that's kind of the basis of the show, because essentially what he's saying here is that it could be a fungi that could emanate, that could maybe get into human brains and control human actions, and then we get the zombies, okay? Now, and there is the zombie ant fungus, which targets ants, obviously, but there is actually another fungus that kind of does that in humans, right? And that fungus is called Cryptococcus neoformans. And it's a fungus that we work with in my lab. It's a fungus that's really interesting because it does some really weird but cool things, right? So it is a fungus, and so it is somewhat dangerous. This is a fungi that actually causes about 180,000 deaths in sub-Saharan Africa every year. But typically, no need to fear. Typically, that's in people who are immunocompromised, right? And typically people who have AIDS and HIV, right? So if you're immunocompromised, you don't want to go near this fungi. It's a fungi that you may have experienced before. If you go hiking in the woods, you might have breathed in some of the spores from this fungi. But typically your immune system can handle it. But if your immune system can't handle it, if it gets into your lungs, if you breathe in the spores, right, what happens is that macrophages within your lungs will try to get rid of that fungi, right? It'll eat those spores, right? It'll ingest it. And once it ingests it, it tries to digest it, okay? But the fungi does something interesting. It actually resists, it's able to resist that degradation within the macrophage, right? It's then able to multiply within the macrophage, right? It's also able to sort of get the macrophage to take it from your lungs to your brain, okay? And then in your brain, it's able to get the macrophage to vomit it back out via vomocytosis, or what's described as called vomocytosis, okay? And so in my lab, we study that. This is a movie taken from my lab demonstrating that. The smaller bead are the fungi, right? Pay close attention here. The smaller bead are the fungi, and you can see that the larger cell is the macrophage, and you can see that at about 14 hours, they start to spit out those fungi from the macrophages. But yeah, this was really interesting to my lab, and this is a behavior that was discovered relatively recently in 2007 by a lab at MGH. So still not a whole lot known about it. And so, I kind of took up the challenge of, how much more can we learn about it? Number one, right? And so we've published a couple different papers looking at, first and foremost, developing tools to study this macrophage fungi interaction, right? Because there weren't these tools before. So we developed a reporter system, a fluorescent reporter system to study when this behavior happens in vitro, right, under the microscope. We developed a algorithm actually to also detect and quantify when and how much, to what extent this behavior happens in situ. And then we also demonstrated that dendritic cells are able to phagocytose. So not only macrophages can vomocytose these fungi, but dendritic cells can also vomocytose this fungi, which brings up some interesting questions as well. But the story, and we're continuing to work on other things in respect to vomocytosis and respect to Cryptococcus neoformans as well. But the interesting story that I'll bring you today is on a system that we developed recently, relatively recently, and that's trying to take advantage of or exploit these bugs for drug delivery to the brain, okay? So I've just told you these things can get to the brain, right? And so we thought, well, we work with particles, right? Why not see if we can put particles on these bugs and see if they can actually shuttle them to the brain? If these particles have drugs, then potentially they can release those drugs and treat different brain diseases, okay? So that's the general synopsis. And why? Well, many drug candidates fail in early stages of development, right? Particularly for CNS diseases, central nervous system diseases, right? So, it's not that these drugs don't exist, right? They're drugs being made, small molecules being made every single day, being identified, isolated every single day to treat, right? Different things, different brain conditions, from neurodegenerative diseases like ALS or Alzheimer's, right? There are things out there, there are drugs out there, right? The problem is that we cannot get them to the brain at high levels, right? Whenever we do clinical trials, what do we look for first? Phase 1 clinical trials. Safety, right? With these drugs that are being designed, yes, they may act in the brain, but they might also act in your kidneys. They might also act in your liver, okay? And so they might have some cytotoxicity there, right? So phase 1 clinical trials, what do we look at? Are these things safe? Okay? So lots of these drugs, they exist. They may not be safe, right? Especially in the concentrations that are being delivered, so that it can be efficacious in the brain. They might cause harm to other organs, okay? Secondly, if they're delivered in lower concentrations, then they don't work, right? So there are drugs out there, but it's more a matter of, can we get them to the site where they're needed to be active to reverse the course of some disease, okay? And getting things to the brain is, getting things to organs, right, is challenging, right? If you take a pill, for instance, right, it has to go through the digestive system, right? So, which means it has to encounter acidity, enzymes, right? Peristalsis, right? Mechanical forces. And then it has to get across your digestive tract into your blood, right? And then in your blood, that's a pretty complex physiological media as well. A number of things it can interact with, okay? Cells as well as different proteins, molecules in your blood. And then it has to get through your liver, right? Which is to decide, okay, should we clear this or not? And then your kidneys, okay? And then if it does end up getting to the cell, right, there are things called MDRs, multi-drug resistant pumps in your cells that can then get rid of those drugs as well. So lots of barriers that we have to get by, right? So the main thing is not necessarily designing the drug. Drug discovery, I think, is in the last 80 years, is relatively easy for scientists. It's how do we get those drugs to the right place, to the right site, okay? And so we're trying to exploit these pathogens to get those drugs to the right site. With respect to getting things to the brain, there's added complexity because of this structure called your blood-brain barrier, okay? And that's sort of this special epithelial layer that covers your brain and ensures that things don't get into your brain. It's very selective. And that's important because your brain is the most important organ in your body. You don't want everything getting there and potentially jeopardizing the communication that your brain provides, okay? Communication responses your brain provides. And so to design things to get into the brain, to be successful, typically we have to follow something called the Lipinski rule. We have to design these drugs that are really, really small and lipophilic. They can get through cell membranes relatively easy. That's not always going to be the case, right? And only 2% of drugs actually meet this criteria, right? So 2% of drugs meet this criteria actually get into the brain, right? And then you have to think of all these other barriers, right? They're getting into the brain. They're probably going to get into your liver as well, kidneys. They're going to be cleared. They're not going to be efficacious or as efficacious as we need them to be. And so that leaves people with neurodegenerative diseases, right? Things like ALS, things like ALS, things like Alzheimer's, Parkinson's, multiple sclerosis, spinal cord injuries, TBI, brain infections, stroke, epilepsy, right? Cancers, glioblastomas, right? And a number of other conditions that affect our brains, right? In a dilemma, right? There's very few things that are available to treat brain diseases, especially your neurodegenerative diseases, okay? And so there is this dire need to develop more effective means of overcoming these barriers to get things to your CNS. And, Cryptococcus neoformans has shown that it can overcome those barriers, right? We don't know how as yet, but it can actually overcome those barriers. And it can overcome those barriers using 3 different mechanisms. In addition to Cryptococcus, these are the other organisms that can actually get to your brain. E. coli is one, C. albicans is one as well. But Cryptococcus is really special in that it can get to your brain using multiple mechanisms. It can get to your brain by getting through spaces between those epithelial cells. I don't mean to scare you. It can get to your brain using what we call transcytosis, right? So actually using receptors on the surfaces of those epithelial cells, right? To then get into compartments within those epithelial cells and be transferred across into the brain parenchyma. Or it can get to the brain, and this is the most used pathway, through macrophages and monocytes. It can allow itself to be eaten by those immune cells, right? Manipulate these immune cells to get to your brain, and then induce vomocytosis so that they can then establish infection within your brain. Okay, so pretty effective cell to get to that gets to your brain. Skip these slides. Yeah, so essentially we hypothesized, what if we could leverage Cryptococcus neoformans, leverage vomocytosis, right, to improve therapeutic delivery to your brain. And to do this, we essentially developed what we call these fungal drug delivery carriers, where we would get Cryptococcus, develop particles that would contain drugs, these packages, right, and conjugate those particles onto the surface of these Cryptococcus cells in the hope that essentially these particles can hitchhike those Cryptococcus cells to your brain. Once they get to the brain, they're vomocytosed into the parenchyma. These particles are biodegradable, right? So they start to break down, they release the drugs, okay? And hopefully they're therapeutic as well. In the interest of time, I'll skip through the science here, but I will show you. So this was recently awarded. This was the 2024 Invention of the Year for University of Florida Technology Innovate Office. And I'll show you that video, and then I'll happily take any questions that you guys may have on this or any other technologies in the lab.
This is a really interesting project. It's a project that I'm super excited about because it is scary in a sense that we are putting bugs, pathogens, fungi into your body to hope to correct brain diseases. People get sick. People suffer from all sorts of diseases. People suffer from immune diseases. People suffer from brain diseases. And we hope to alleviate the suffering of these people. We hope to extend people's lives. We have a plethora of drugs that work effectively against them in the lab, right? And in animals, right? And that's because we can inject those drugs directly into the brain or into the spinal cord. In reality, though, in humans, we can't inject, or it's scary to inject something into the brain directly or into your spinal cord directly. What happens is that these drugs, because they're injected into your veins instead, they're not delivered into the brain at high levels. And so what we're trying to do is utilize this fungi that has a natural attraction, a natural tropism for the brain, and put drugs that would treat some of these brain disorders in particles, attach those particles to these fungi, and then hope that once we inject those fungi, they then go to your brain. The drugs can be released from those particles and treat disorder that is impacting the patient. This is biotechnology. It takes a really long time to translate into humans. But in 10 years at least, I hope that we're at least at clinical trial stage. My hope is that we can really push this from the lab to animals and into humans. And certainly at 10-year anniversary, we are doing our first in-human, or even before that, our first in-human clinical trial. And the biggest acknowledgements really and truly go to my students. And that's particularly to Mr. Clinton Smith, who is sort of the main driver of this project, and one of my past students, Dr. Noah Pacifici. And having this award being given to us, it really fuels and drives our ambitions to really take this into the clinic. It really pushes us and encourages us and gives us a sense that this is something real, this is something that has possibilities to really impact human health. And like I said, it'll drive us even more to really fulfill the potential of this idea.
The invention that Jamal and Clinton came up with is
Stop it there. I'll stop it there and first and foremost thank my lab. I don't do any of this work. It's all done by my trainees, postdocs, and grad students, and undergrads actually. And as well as funding sources, and I'm happy and collaborators and happy to take any questions you guys may have. Thank you.